Boleh dengar saya tak? Assalamualaikum.
Waalaikumsalam, Doktor.
Okay, good morning, Danu. Okay. Okay, while waiting for your friends, baru 31. First of all, sorry for the last minute notice. I have emergency this morning, so for today we're going to have lecture as a virtual lecture, okay, online class. Okay, hopefully by Wednesday we can meet. We can have a physical lecture, okay, next Wednesday, and in the afternoon we're going to have a lab class, okay. So apa khabar semua pagi ni? Dah breakfast?
Belum, Doktor.
Belum. 9 o'clock belum breakfast. You all breakfast pukul berapa?
11.
11 tu bukan breakfast. Itu dah lunch. Okay, I just uploaded the lecture notes on nucleotide metabolism. So today, our topic would be nucleotide metabolism. Okay, so when we talk about metabolism, I hope you understand it involves biosynthesis process, degradation process. Okay, as you know that what does it mean by metabolism? It involves anabolism, it involves catabolism. So, all right. In the middle of the class, at 10 a.m., saya akan ambil attendance lah biasanya. Okay. Okay, let me share screen. Okay, can you see my screen? Let's wait for another. So hari ni banyak lecture ke? Banyak kelas?
Ada banyak, Doktor.
Banyak juga, okay. I hope you can get ready by having your notes ke apa tu piece of paper for you to write down any important points that I will share with you. Even though you have the notes, but sometimes I'm going to share. Okay, I just want to emphasize here. Lecture notes is only as a guidelines. Okay, more explanation. Dia sangat brief. Okay, so that's important of having lecture because every single points in the lecture notes will be discussed. Okay, will be discussed in details. So you may get extra explanation, deeper explanation during lecture. So kat situ you may jot down important notes. Okay, that may useful for your exam, that may useful for your daily life. Okay, sebab as a biochemistry student, basically what you learned, okay, during these 4 years is basically something that you can relate, something that you can apply to yourself or to your family or to the community. So basically, apa sahaja yang you dapat from the lecture, contoh you belajar tentang carbohydrate kan, okay, you may share your whatever you get from the lecture with your family. Kan? If you learn about food biochemistry, maybe we can discuss on that, share with it with your friends ke apa kan. So that's important of having of learning biochemistry. Okay, it's really relatable. It can be applied, okay, in your daily life. Okay, so day 40. Okay, let's start. Okay, this is our first topic. Okay, as I'm taking over from Dr. Dina. Okay, Assalamualaikum warahmatullahi wabarakatuh and a very good morning. Okay, so we start with nucleotide metabolism. Okay, I hope all of you, kalau yang can join our first meeting last week, so I hope you know me. Okay, you can call me Dr. Adila. Okay, I'm going to take over the second part of the BCH 3110. Tak silap saya, I'm the post coordinator. Okay, saya sudah saya. If you have problem and any problem with the subject, okay, you can discuss with me. Okay, you can text me, you can email me, or you can set an appointment to see me. Okay, anything related to subject. Okay, if you have other problem pun tak ada masalah juga lah kalau nak jumpa saya. Okay, so when we talk about nucleotide metabolism, so this is the learning outcome that what we would like to achieve, okay, at the end of the topic. Okay, so first of all, you should be able to distinguish and draw nucleotide structure. I'm sure you know what is nucleotide. Saya dengar kan, what is nucleotide? And now as a biochemist student, you should be able to distinguish, you should be able to draw nucleotide structure. Okay, then you have to be able to explain and understand the function of nucleotide. Okay, so in general, you know what is nucleotide. Okay, so you know the importance of nucleotide, but later we are going to discuss in details. Okay, what is what are the functions of nucleotide? Okay, why is it important to our body, especially to the metabolism of protein and nucleic acids? Then we move to the pathway. Okay, so we have this de novo meaning that it's a new pathway. So it's an anabolic pathway. Okay, we want to build up new molecule. And also salvage pathway. Salvage pathway meaning that is a pathway to synthesize new molecule, but it utilizes degraded product. So it utilizes degraded product. Okay, so that all the degraded products is not going to be something that wasteful. Okay, so it's going to be utilized by this specific pathway to synthesize a new molecule. And also the most important thing is you should able to show your understanding, okay, on the catabolism process of purine and pyrimidine. So basically in this topic, we are going to discuss in details about purine and pyrimidine metabolism. Okay, so in bigger picture, okay, in nucleotide metabolism we are going to look on introduction. So what is nucleotide? What is nucleotide metabolism? Okay, what is the role of this metabolic nucleotide? Okay, function dia apa? What is the importance, okay, of nucleotide? Then we are going to discuss on the metabolism itself, on the synthesis process, and also degradation process of nucleotide. Okay. Okay, so to start, okay, on the nucleotide metabolism. So I give you this overview, okay. So I'm sure you know what is central dogma. So anybody want to describe what is central dogma? What do you understand with central dogma by looking at this diagram? Anyone, what is central dogma? Biasa dengar kan central dogma, I'm sure you've heard this. Biasa dengar tak central dogma?
Pernah dengar.
Okay, what is central dogma? Okay, you have this diagram, contoh kan diagram ni. Okay, from DNA to RNA to protein. So how do you describe? Okay, in brief explanation, describe what is central dogma. Anyone?
Central dogma is a process where DNA is turned into functional protein.
Okay, DNA turning into functional protein. Thank you, Adeline. Okay, what else? How do you describe? So that's another, that's one of the description of what is central dogma. Okay, how about other description? How do you define central dogma? Kita boleh, by looking at that diagram, you can give your own description based on your understanding. Okay, contoh. Okay, behind that, okay, DNA into functional protein. So that one. Apa lagi? What else? Okay, how do you describe what is central dogma? Okay, the process of making protein from DNA. Okay. Okay, transcription, translation. All right, correct. Okay. So in general, we can say that it explain, okay, is the explanation of flow of genetic information within a biological system. So that one is a very general. So it tells us about the flow of genetic information. We know that our genetic information contains, okay, contains in DNA, okay. So in DNA, it contains the instruction to make a protein. So RNA copy all the instruction from DNA, and later it being translated into the functional protein by process of translation. So you can describe it in many ways, okay, as long as it explain about importance of central dogma. Okay, so this is your central dogma. So how about the diagram on your right? Okay, so this is your DNA. Okay, DNA structure. Okay, so by looking at this GCUA, you know that's a basis. Cytosine, uracil, guanine, adenine. So we are going to look at this the structure of nucleotide in the next slide. Okay, so one thing that you should highlight is the importance of central dogma. Is we know that DNA the process is having store and transmit genetic information from one generation to the next. Mental process in our daily life. Contoh, okay. What kind of genetic information that you carry from your parents? Anyone? That you think that can be inherited from your family, from your father, from your parents? Anyone? What kind of genetic information? Contoh.
Height,
Sorry?
ketinggian,
Ketinggian. Okay, what else? Genetic information. Contoh. If you have blue eyes, that one basically you can inherit from your parents. Okay, let's say your parents one of your parents akan ada blue eyes kan, mata biru, so you may get that. Okay, so that information, okay, is inherited, okay, from your parents and can be translated to you. Okay, so that you pun akan dapat mata biru. Contoh apa lagi? You punya rambut perang. This is original, a very yellowish hair kan, and this one is bukan yang buat-buat. Okay, so that kind of genetic information is something that you can inherit, okay, from your parents. So this important information contains in DNA that can be inherited, okay, that can be translated into protein. Okay, so that you will get, okay, whatever protein, okay, you will all the protein will be exhibit. Okay, kalau you dapat protein mata biru, so you will exhibit, you akan dapat mata biru. Okay, so that kind of genetic information. Okay, blood type, disease, probably yes. Okay, contoh cancer cells. So basically, in general, most of us has that cancer cells, but maybe yang membezakan kita akan dapat cancer cells or tidak adalah it is activated, it is being induced, okay, and that one it involves many other factors, contoh environmental factors, your diet, okay, so that kind of environmental factors will contribute, okay, to the activation of certain gene, contoh cancer ke apa ke. Okay, blood type, yes, blood type, correct. So you can inherit that. Okay, contoh kalau your parents has blood group A, okay, you may get blood group A, but you may get O blood group as well. It depends kan, dia punya diagram to show from genetic information what kind of blood group that you can get. Okay, tak sebenarnya kalau parents you B, you dapat B. Okay, you can still get O. Even my parents B, my blood group is O. So so it depends on the information, okay, on the inheritance of the genetic, okay, on the gene. Okay, all right, so this is DNA. Okay, so by looking at this diagram, you can see that DNA structure is a very important finding. Okay, we know that it has so many important roles. Okay, it contains all the genetic information, so that semua, okay, all the information, okay, that required, that can be translated, referred to as a gene. Okay, gene mata biru, gene rambut perang, gene for cancer cells, gene for many things. Okay, we have million of genes. Okay, million of genes. Okay, you can do your own reading on what is gene. Okay, what is gene? All right. So you tahu juga kan, kalau dalam in molecular biology we have this genetic code that being translated into three coded genetic code. Okay, a PG as a start codon. Okay, so that's how gene or genetic code being translated in a triplet coded. Okay, all right. So what is nucleic acid metabolism? So it's a process by which nucleic acids are synthesized and degraded. Okay, synthesized and degraded. Basically, it involves, it explains to you on the anabolic and also catabolic metabolism. Okay, so nucleic acids are polymers of nucleotides. So nucleotide synthesis and anabolic mechanism generally involving the chemical reaction of phosphate, pentose sugar, and nitrogenous base. So basically, these three molecules they explain to you on the structure of nucleotide. Okay, so this is nucleotides. So if it need to contain phosphate, okay, it need to contain pentose sugar and also nitrogenous base. So if one missing, it is not known as a nucleotide. So it has to contain all three, okay, to form a nucleotide: phosphate group, sugar, and also nitrogenous bases. Okay, basic kind. Okay, I'm sure you know about the structure of nucleotides. Okay, so this structure of nucleotide, okay, phosphate, I'm sure you know phosphate punya structure. Okay, how do you draw phosphate? Okay, and we have this pentose sugar. Okay, when we talk about pentose, you know that how many carbons it has. Okay, pentose sugar, either it exists as a deoxyribose for DNA or ribose sugar for RNA. Okay, ribose or deoxyribose. Okay. All right, and also we have this nitrogenous base. So nitrogenous base, it has 4 different nitrogenous base. We have guanine, cytosine, adenine, thymine, and uracil. So uracil is for RNA. Okay, only exists in RNA. So by looking at this structure of the nucleotide, so you need to know that the phosphate group, okay, attached. Okay, how does it link to the pentose sugar? And where is the location of the attachment of nitrogenous base to the pentose sugar? So basically, phosphate and nitrogenous base are attached to pentose sugar at their specific location. Okay, and in DNA and DNA structure, phosphate and sugar make up the backbone. So this is the important attachment that give us the structure of DNA. The double helix structure is supported by this backbone between phosphate and pentose sugar. Okay. All right. Ada soalan? Okay. You can just you can interrupt me anytime if you have a question. Okay. Okay, so if we look at the name, okay, nitrogenous bases, it tells us that it's a basis that contain nitrogen in the structure. Okay, what does it mean by nitrogenous bases? It's a basis that contain nitrogen in the structure. Okay, now look at the structure of all these bases. Okay, so nitrogenous bases can be classified into 2 groups. One is pyrimidines and another one is purines. Okay, pyrimidines and purines. So how do you differentiate between pyrimidines and purines? Okay, by looking at this diagram, how do you differentiate purines and pyrimidines? Anyone?
Ring structure.
Okay, so we can differentiate, first thing first, we can differentiate in terms of structure. So purines, okay, it has 6-membered nitrogen containing ring that fuse with to an imidazole ring. So basically purine, it has 2 ring structure, okay, 6-membered nitrogen containing ring that fuse to the imidazole ring. So that one for purines, okay. But for pyrimidine, it only contains 6-membered nitrogen containing ring. Okay, yang membezakan purine and pyrimidine. But in cells, purines and pyrimidines can be found in equal amounts, okay, in cells, okay, either for purines or pyrimidine, because we know that in DNA, nitrogenous bases, it exists in a pairing, kan, as a pair, okay, between ATGC. Okay. Contoh, okay, it's a pairing between purine and pyrimidine. So that's explain the equal amount of purine and pyrimidines that present in cells. Okay. Okay, number of ring based on rings. Okay. All right. So, okay. And I want you, okay, by looking at the best. Okay. How do you, can you name, okay, let's say for cytosine. Okay, can you name the structure of the, so pyrimidines, okay, it consists of cytosine, thymine, and uracil. So uracils only exist in RNA, but for purines, we have this adenine and guanine. Okay. So basically for bases, natural bases is an aromatic heterocyclic ring. Okay, aromatic heterocyclic molecule, okay, that belongs to either pyrimidines or purine. And purines, we have this adenine and guanine. Okay, so we have, okay, kalau nucleoside, okay, bases and sugar will form nucleoside. Okay, and this nucleoside play an important role as a precursor to the formation of nucleotide. So nucleoside, when is attached to the phosphate, combined with phosphate, it will form nucleotide. Okay, so you need to know the difference between nucleoside and nucleotide. Okay, all right. So, okay. Okay, how phosphate group, okay, are linked to the pentose sugar. Okay, so the linkage is through ester bond. So phosphate group attached to the carbon 5 of ribose sugar through the ester bond. Okay, but for nitrogenous bases, okay, how they attach to the pentose sugar, okay, they attach at the carbon 1 of pentose sugar. But the attachment is different between pyrimidine ring and also purine ring, where the pyrimidine ring, the N1 of the pyrimidine ring get attached to the ribose sugar. But for the purine ring system, N9 get attached to the ribose sugar through glycosidic bond. Okay, so that's the difference between pyrimidine and purine in terms of the attachment to the ribose sugar 1 through N1. For the purine is through N9. Okay, and the attachment is catalyzed by the glycosidic bond. Okay, for the formation of the backbone of DNA, the attachment of phosphate to the ribose sugar is achieved through ester bond. Okay, so dekat sini, okay, for you to understand on the structure of nitrogenous bases, okay, you should be able to identify, okay, the numbering of the structure so that you can understand. So numbering, kalau purine, we start from here. Okay, 1, 2, 3, 4, 5, 6. Okay, but is different from the purine system where the N1 starts from here, N1, then carbon 2 and 3, carbon 4, 5, 6, and 7, 8, and 9. Okay. Okay, ada soalan? Okay, by looking at the structure, okay, of purines and pyrimidines, let's describe the specific name, okay, the chemical name, okay, the specific name of adenine. Okay, for example, I will give, okay, saya bagi contoh untuk adenine. Okay, so adenine, or we can name it as a 6-amino purine. Okay, 6-amino purine. Okay, selain pada adenine, we can name it as 6-amino purine. So that we know that adenine structure contain amino group at carbon 6. Okay, how about guanine? I think nak try. How about guanine? Anyone? Okay, how about guanine? 2-amino purine. How about carbon 6? Okay, 2-amino and 6. How about 6? Okay, you have 2 here at carbon 2 and also carbon 6. Okay, 2-amino and 6 deoxypurine. Okay, so that one is for guanine. How about uracil? How about uracil? Two, four, tapi bukan dihydroxy. Hydroxy meaning OH. Okay, so this one is dioxy. Okay, 2,4-dioxy pyrimidine. How about timing? You have 1, 2, 3. How do you describe that? Oh, tak nak buka mic ke? How about timing? Let's see, kalau dalam exam saya bagi nama specific name kan, so that you know, okay? Saja nak speak here and there. Okay, 2,4-dioxy 5-methyl. Okay, 5-methyl. So this is a methyl group. 5-methyl pyrimidine. Okay, so that one is for thymine. How about cytosine? You have this. 2-dioxy and 4-amino pyrimidine. So that one for cytosine. So that's how, okay, apart from the general name, adenine, guanine, uracil, thymine, and cytosine, by looking at the structure, you can name, okay, you will understand the structure of adenine by looking at the functional group that it contains. Okay, contoh adenine, dia ada amino group carbon 6. Okay, and for example, guanine, it has amino group and also it has this dioxy functional group, so that you understand, okay, the difference between the two purines, adenine and guanine. Okay, contoh adenine dia tak ada dioxy. Okay, even though both have amino group, but adenine dia tak ada dioxy group. Okay, how do you differentiate uracil, thymine, and cytosine? Okay, so thymine paling senang nak ingat, it has this methyl group. Okay, and 2-dioxy group. Okay, and but for uracil dia beza dengan thymine. Okay, dia tak ada methyl group. Okay, cytosine a bit different because it contains amino group. Okay, and 1-dioxy group. Okay, ada masalah? At least I want you to understand the structure. Tak naklah tengok, okay, so what is this? That's it. Okay, so that you able to differentiate, okay, the structure of adenine and guanine, so that you do understand what are the functional group that each of the nitrogenous bases have, okay, and what makes the difference between the structure of nitrogenous bases. Okay, alright, boleh? Okay, selain pada that 2 rings and 1 ring, so that you understand about the structure of the nitrogenous bases. Ada soalan? Saya rasa kelas lecture yang paling senyap kelas you all lah. Kalau sepanjang saya buat online selalunya bersahut-sahutan. You all banyak tulis dekat chat. Okay. Alright, saya kadang tak sempat nak buka chat. Okay. Alright. Okay, so now let's look at the nomenclature, okay, on the difference between nucleoside and nucleotide. So in the previous slides, okay, we have discussed on the difference in terms of the structure, the difference between nucleoside and nucleotide. Okay, nucleoside, we know that it only contains nitrogenous base and also pentose sugar. Okay, a bit difference between when you compare with nucleotide, and we know that nucleotide has that 3 components. Okay, phosphate, nitrogenous bases, and also pentose sugar. In terms of naming, okay, between nucleoside and nucleotide, so dia pun berbeza lah, even though for RNA and for DNA, kalau DNA dia ada it named as a deoxy. Okay, kenapa deoxy? Why deoxy? Why DNA, deoxyribonucleic acid? Okay, it has that deoxy. Kenapa dia ada deoxy? Siapa tahu? I'm sure you tahu. In terms of structure. Kita pergi structure. Siapa yang jawab ni? No oxygen. So no oxygen dekat mana? Deoxy, no oxygen dekat mana? Okay, carbon 1. Okay, so it tells you on the structure of the presence of oxygen. So there is no oxygen, okay, at the carbon 1 in the structure of DNA. So that's why the name of DNA, it has deoxy. Okay, so RNA it is known as adenosine, okay, for adenine, but for nucleotide it's a adenylate. So you should be able to differentiate between, kalau saya bagi dia adenosine, you automatically know this is a nucleoside. Saya kata, okay, right, draw a structure of guanosine, for example. So, okay, so you should be able to identify whether either it is a nucleoside or nucleotide. Okay, if it is nucleoside, you should know that it only contains ribose sugar and also nitrogenous base. Okay. Okay, so this is structure of pentoses. Okay, your ribose sugar in terms of DNA. Okay, this one for DNA, RNA. So kat sini tak ada kan? Okay, that's why deoxy. So kat carbon 1 is only has hydrogen. Okay, but for RNA it has this OH. All right. Okay, so now let's look on the phosphodiester linkages. Okay, so phosphodiester linkages, we know that this is the link that attach phosphate group to the pentose sugar. Okay, so it makes up a DNA backbone. Okay, so this is the phosphodiester bond that link. Okay, that link two nucleotides together to make a backbone. Okay, to make a backbone. For example, this is your DNA. Okay, this is your DNA. Okay, you have this nitrogenous base, you have this pentose sugar, and you have this phosphate. How do you link to another nucleotide? Is by this phosphate is attached to the pentose sugar of the new nucleotide. Okay, so it is linked by phosphodiester. Okay, ester bond. Okay, phosphodiester. All right. So you know, okay, you can see the attachment link occurs at the carbon 5 pentose sugar to the okay, this 5 carbon, okay, 5 carbon 5 of pentose sugar attached to the phosphate sugar. Okay, phosphate sugar. And this phosphate sugar, this phosphodiester linkage, okay, that link to the next nucleotide at the carbon 3 of the pentose sugar. So that's why the DNA sequence is right, okay, from 5 prime to 3 prime. Biasa dengar tak? Okay, 5 prime to 3 prime. So that's how DNA sequence is reading. Okay, the reading of DNA is from 5 prime to 3 prime. It based on the to the link of nucleotide units. Okay, sebab dia punya attachment from berlaku dekat 5 prime or carbon 5 of the first nucleotide and at the 3 prime of the second nucleotide. So that's how. So that's why kalau kita tulis DNA sequence, okay, is from 5 prime to 3 prime. Kita tak tulis DNA daripada 3 prime to 5 prime, or kita tulis DNA from 1 prime ke 6 prime ke tak. So why 5 prime? Dia tengok kat mana attachment of nucleotide berlaku. So that's how we read DNA. Okay. Alright. Okay, so selain besides phosphodiester, besides ester bond, okay, that link nucleotides unit, so we do have hydrogen bond. Okay, hydrogen bond that link two nitrogenous bases together. Okay, we know that nitrogenous bases exist as a pairing. Okay, adenine and thymine. Okay, and also guanine and cytosine. Okay, so it is understood as a pairing. Okay, dia kena A dengan T, C and G and C. Okay, in general, hydrogen bonding is not that strong compared to ester bond, but collectively, let's say we do have this hydrogen bonding dalam structure of DNA that link, okay, this hydrogen bonding that link your nitrogenous bases from one DNA strand, that link these nitrogenous bases of one DNA strand to the nitrogenous bases of the second DNA strand. Okay, so that's why kita akan ada double helix, okay, because of the pairing of this base nitrogenous base. Okay, so even though hydrogen bonding is not that strong compared to ester bond, okay, but collectively, okay, when it make up DNA, okay, this double helix DNA, it is considered a very strong hydrogen bond. Okay, secara collectively. Okay, when it make up a DNA double helix structure, so this hydrogen bonding becomes a very, very strong bonding. Okay, and by looking at this diagram, you know that A, adenine and thymine, it contains two hydrogen bond. It is bound by two linkages. Okay, kalau G and C, it has these 3 hydrogen bonding. Okay, that link between guanine and cytosine. Okay, ini ada kita 1, 2, 3, but for adenine and thymine, it has 2. Okay, so basically this is the pattern for the hydrogen bonding that has been defined by Watson and Crick. So they are the scientists that discover about DNA structure, the double helix structure. Okay. Okay, points to remember. Okay, DNA stands for this. So basically, these first few slides just give you an overview. So let's refresh back about the structure of nucleotide. So what are the components involved? So it has pentose sugar, phosphate group. Okay, we have this nitrogenous bases. Okay, we have these purines and pyrimidines. You already understand the structure. So what are the functions group that make a difference between purines and pyrimidines? Okay, so okay, there are two condensation reactions occur when the base and sugar form a glycosidic bond. Okay, that one is base and sugar. And for phosphate and sugar, it will form an ester bond. Okay, so this phosphodiester bonds, okay, so it will build up a backbone for DNA, okay, with the bases all facing inwards, okay. Okay, the two strands of the DNA molecules form antiparallel, okay, so you know what is antiparallel, okay. One strand runs 5' to 3', and the other strand runs to 3' to 5', okay. And the strands wind around one another into a double helix, okay, like a twisted ladder. So this is your double helix DNA structure. So one strand, okay, runs from 5' to 3'. So another strand runs from 3' to 5', so that it complement each other. Okay, it complement each other, and all the bases from one strand dia akan pairing dengan bases with another strand of DNA. All right, ATGC. Okay, and the linkages. Okay, the linkages of two of the nitrogenous bases. Okay, this occurs by hydrogen bond. Okay, so let's look on the function of metabolic nucleotide. So kita dah, we have finished with the introduction on the what is nucleotide, okay, what is pentose sugar, what is nitrogenous bases, okay, what is DNA. Okay, now let's move to the function of nucleotide. Okay, so selain daripada it stores genetic information, nucleotide has other important functions. Okay, the main one is as building blocks of nucleic acid. Okay, for the RNA, it stores genetic information. Okay, for RNA, it copies all the information from DNA and translated into protein. Okay, it also acts as a precursor of RNA. Okay. Also, what does it mean by precursors of RNA? We will discuss later. Okay. So nucleotide also act as a carriers of chemical energy. So it has a role, okay, of ATP and GTP, okay, for the energy production. Okay. And also nucleotide do also involve, okay, act as a cofactors and coenzymes. So what are cofactors and coenzymes? Okay, okay, it involves, okay, cofactors, coenzymes, contoh NAD+, your NADH, FAD, apa lagi. So nucleotide contribute, okay, to the formation of cofactors and coenzymes. And also nucleotide important in the signaling process. Okay, so it act like hormones, okay, or transducers of the information. Okay. Okay, so let's move. Okay, let's discuss in details what does it mean as of a Golgi block. So this is the most important point. Okay, the role of nucleotides in biochemistry. So for building blocks of nucleic acids and RNA, we know that let's discuss in RNA in RNA in specific. Okay, we know that RNA it has three different types of RNA. We have ribosomal RNA, we have mRNA, messenger RNA, and also we have tRNA. I'm sure you know about these three types of RNA. Okay, so let's just refresh about this RNA. So it plays an important role in the central dogma. So basically, it involves in central dogma juga lah. Okay, your ribosomal RNA. So ribosomal RNA is a component of ribosome that very important in the synthesis of protein. Okay, mRNA, we know that is a messenger that carry genetic information from one gene to a ribosome. Okay, and also transfer RNA, we translate. Okay, this vector molecule that has to translate the information in mRNA into a specific sequence of amino acids. Okay, second one. Okay, how nucleotides contribute, okay, in the chemical energy in cells. Okay, so we know that we have phosphate group, okay, in the structure of nucleotide. Okay, then we have contoh. Okay, let's see for bases adenine. Okay, adenine when it has base, okay, one phosphate known as adenosine monophosphate. It has two phosphate group is known as adenine diphosphate. It has three phosphate group is known as adenine triphosphate. So basically, indirectly, secara direct lah, okay, all these nitrogenous bases, all these nucleotides involved in the chemical energy production. Okay, okay, contoh guanine. Okay, I'm sure kita dah jumpa kan GTP dalam TCA cycle. Okay, instead of ATP, it produces GTP. Okay, if you still remember the TCA cycle, then we have this cytosine. Okay, we have this CTP. Biasanya you akan jumpa dekat lipid metabolism, if I'm not mistaken. Okay, and uracil as well. Okay, uracil mungkin dalam protein. Okay, I can't remember. Okay, but for deoxyribonucleoside, we have this ATP, GTP, DCTP, DPTP. So this how, okay, depends on the how many phosphate molecule. Okay, kalau satu jadi mono lah, mono di or triphosphate. So how does it make this nucleotide, okay, as an energy carrier? Okay, you know that phosphate group is attached to the ribose sugar through phosphodiester bonds. We know that phosphodiester bond is a very high energy bonding. Okay, contoh itu baru satu. Let's say you have this triphosphate, so it has a very high energy bonding. So for example, for ATP, GTP, ATP, especially yang ada 3 phosphate molecule. So if you have these 3 phosphate molecules, so it shows that it has a higher when the bonding is being breakdown, it will produce a very high energy, okay, that can be used by cells. Okay. Okay, so this how this, okay, the hydrolysis, okay, the breakdown of the ester bond, okay, anhydride bond, okay, ester bond or anhydride bond, it will produce more energy. Contoh kalau monophosphate, okay, triphosphate obviously will produce more energy compared to monophosphate because of the it has 3 ester bond. Okay, so that's how nucleotides contribute to the chemical energy in cells. Okay. Okay, so nucleotide also involve also act as a component in many enzyme cofactors. Okay, contoh apa? Enzyme cofactors, coenzyme. We have this coenzyme A. Okay, we have this NAD+, NADP, FMN, okay, FAD. So all this, kalau you tengok, okay, NAD is okay, nicotinamide adenine dinucleotide. So basically, adenine is a nitrogenous base. Okay, so kalau you tengok structure, it has few space. Okay, nicotinamide adenine dinucleotide. Okay, dia ada dua nucleotide kat situ. Okay, this nicotinamide dia attach pada adenine dinucleotide. Okay, so this is your nucleotide basically. Your NAD is a nucleotide. Okay. Contoh, okay, this one coenzyme A. Coenzyme A, okay, even though the name is coenzyme A, but in the structure of coenzyme A. So this is the structure of coenzyme A. Okay, kita sebenarnya kan in lipid metabolism, we are going to discuss on the structure of coenzyme A. Yes, okay, but we are going to do some comparison with apa yang saya tak ingat. Okay, so if you look at the structure of coenzyme A, okay, panjang structure dia, it has this mercaptoamine that attached to this pantotenic acid, okay, and that link to the 3-phosphoadenosine diphosphate. So this is your nucleotide. Okay, this is your nucleotide kan. You ada phosphoadenosine diphosphate. So this is your nucleotide that attached to the pantotenic acid and mercaptoamine. So it forms, this nucleotide forms coenzyme A. Okay, so that's how nucleotides act as a precursor for many enzyme cofactors. Okay, later, okay, if you have time, okay, go and look for the structure of AD. FAD. So, kalau you tengok AD ni kan, of course lah, adenine diphosphate ke, okay, or adenine dinucleotide ke, okay, go and look what does it mean by FAD, FMN. So where is the nucleotide dalam structure FMN, FAD, okay, boleh, okay. Okay, so this one is for signaling, how nucleotides involved in signaling. Okay, so these are the three regulatory nucleotides. We have this CAMP, cyclic AMP. So AMP is stand for adenosine monophosphate. So adenosine monophosphate, you know that it is a nucleotide. Kalau tengok structure dia, okay, dia ada base, dia ada sugar, dia ada phosphate. So it is nucleotide. So in signaling, we have this CAMP, CGMP, okay, and also GPP, okay. Bila belajar signaling dalam lipid metabolism, akan cukup banyak, CAMP, CGMP, alright. Okay, ada soalan? You want to have a break for 5 minutes before we continue with metabolism? Hello you all. Assalamualaikum.
5 minutes.
Okay. Okay, let's have a break for 5 minutes before we continue with the metabolism process. Okay. You all ada lagi tak kat belakang ni? Sebab tak nampak kan?
Ya.
So saya cakap seorang. Ada. Okay, 10:05 lah, kita jumpa. Okay? Okay. Sana saya ambil attendance. Attendance. Saya stop sharing sekejap ya. Ramai ni yang tak ada. Tambah sana. Okay. Okay. Mau lagi minum? Mari kita mula.
Yes, right. Boleh.
Okay. All right. Okay, now, we have discussed on the introduction part. Time, nutrition spaces, pentose sugar, and also phosphate group. Okay, and also we have looked at the function of nucleotide. Okay, now let's discuss on the metabolism of nucleotide. Okay, so in general, there are two different pathways involved in the synthesis of nucleotide. Okay, belum degradation ini baru synthesis, but there are two different pathways. One is de novo synthesis. De novo meaning new. De novo, new. Okay, so it begins with their metabolic precursor, with their template. Okay, for example, amino acid, ribose, biphosphate, carbon dioxide, and one carbonate. So it tells us that the de novo synthesis, the process starts from scratch, and it requires a simple precursor molecule for the biosynthesis process to occur. So that one is de novo synthesis. But for salvage pathways, is a bit different compared to it. The synthesis direction utilize the free bases, okay, or degraded products or nucleotides related from nucleic acid breakdown. Okay, so basically utilize the degraded products, okay, to synthesize nucleotide. Okay, so it's synthesized from intermediates in the degraded pathway for nucleotide. So the goal is similar. Okay, so you want to synthesize nucleotide, but it can be done in two different ways. One from scratch, the de novo synthesis, one through salvage pathway, which utilize the degraded product, degraded intermediate, okay, to synthesize nucleotide. Okay. Okay, so de novo pathways. Okay, so now let's focus on de novo pathways. So for purine and pyrimidine biosynthesis, so the de novo pathway is identical, okay, similar. It's basically all the reactions similar in all organisms. Okay, but what makes the reaction is different because we know the structure of purines and pyrimidines are different. So purines is two-ring structure. Okay, so it contains six-membered nitrogen containing ring that fills the middle ring. Okay, compared to the pyrimidine. So for purine bases, basically dia punya structure are already assembled and attached to the ribose ring. But for pyrimidine, it is synthesized as an orotate. So basically, orotate is an intermediate in the biosynthesis of pyrimidine. So we are going to look at the pathway that involved in the synthesis of pyrimidine. So, okay, so bases are not intermediate in the de novo pathway. Okay, kita tahulah. Okay, what is bases? So all the adenine, guanine, cytosine semua tu, it is not intermediate. Okay. So several important precursors are shared by the de novo pathway for the synthesis of pyrimidines and purines. So basically, we are focusing on the synthesis of purine and pyrimidine. Okay, so this is the PRPP. Okay, 5-phosphoribosyl, 5-phosphoribosyl, 1-pyrophosphate. Okay, PRPP. Okay, so this is a precursor. Okay, important precursor for both purine and pyrimidine synthesis. Okay, so in the photosynthesis pathway, amino acid plays a very important role as a precursor, either in purine formation or pyrimidine formation. For example, glycine, okay, in purines, aspartate in pyrimidine. Okay, also aspartate also contribute in the purines formation. Okay, and glutamine also acts as a source of amino groups. Okay, kena tahu kan you have been discussed on the what is amino acid, ada berapa amino acid, in terms of the grouping of amino acid, you have learned that. Okay, so you should know. Kalau sebut kat sini glycine, at least you can recall. So how does the structure of glycine looks like? Is it an acid type of amino acid? Is it a basis, or is it has this specific functional group? Okay, how about aspartate and also glutamine? Okay, glutamine it contains amino groups, so that's why it acts as a source of amino groups for the synthesis of nucleotide. Ingat lagi tak structure glycine, aspartate? Okay, so later you should start to look at the structure, okay, of each intermediate involved in the synthesis of nucleotide. Okay, all right. Okay, so purine. Okay, purine, we know that it has two rings. One is six-membered nitrogen-containing ring, and one is imidazole ring. So kita dah tengok kan structure tu macam mana. And basically this is where the sources of each component in the purine structure. Okay, for example, okay, if you still remember, for purine, the attachment of purine bases to the ribose sugar occurs through N9. Okay, and we look, okay, the sources of the N9 is from ribose phosphate. Okay, so ribose phosphate is the first intermediate, okay, in the formation of nucleotide synthesis. Okay. Okay, so N9. So let's look at N1. So N1 is contribute from aspartate. Okay, so carbon 2 and also carbon 8 is contribute from tetrahydrofolate. Okay, and 3 from glutamine. Okay, carbon 4, carbon 5 is from glycine. Okay, carbon 6 from carbon dioxide. So this is how it shows us the sources of each component in the purine nucleotide structure. Okay, in the purine ring structure. Okay, there ada daripada amino acid, okay, from glutamine, from aspartate, there is from glycine, okay, from tetrahydrofolate, from carbon. Okay, so later, once it forms this purine, so it undergo reactions to form inosine monophosphate before it can break down into ATP or GTP to RNA. Okay, so this is the origins of the atoms in the purine ring. Okay, but but in the reactions involved, it has the sequence, okay, on the formation. Kat mana dia bukan from N1 ke dulu ke apa, okay? There is a sequence on the formation of purine structure. Okay, so so this is another summary on the sources of atoms, okay, in the purine, okay, from aspartate format, okay, 2 and 8 from format or tetrahydrofolate. Okay, tengok ada certain books yang guna tetrahydrofolate, ada certain books yang guna format. Alright, and N3 and N9 from glutamine, carbon 4, 5 and 7 from glycine, and carbon 6 from ada yang guna carbon dioxide, ada yang guna bicarbonate. Okay, do not make yourself confused. Okay, so depends on, as long as tak jauh lah. Okay, tetrahydrofolate and format. Why is it some book purposely use format instead of tetrahydrofolate? But okay, we can look at the structure of it between the combination between format and tetrahydrofolate. Same goes to bicarbonate and also carbon dioxide. Okay. Okay, so aspartate format, glutamine, glycine, and bicarbonate acts as the building blocks for purine synthesis. Okay. All right. So this is the reactions. Okay, for the purine nucleotide synthesis that begins with PRPP. So basically, I think this reaction, this pathway, kalau dalam diagram ni lah, I think it missing one, the first step. Ada buku yang terus step PRPP, but I just want to add here, at least you can see the link, okay, in the formation of purine nucleotide. Okay, we have this ribose 5-phosphate. Okay, the formation of PRPP is from ribose 5-phosphate. Okay, ribose 5-phosphate. Okay, it react with ATP, so there is a transfer of phosphate group to the ribose 5-phosphate, so it forms 5-phosphoribosyl pyrophosphate. Okay, PRPP react with ATP. Okay, so it forms PRPP. Okay, for 5-phosphoribosyl pyrophosphate. Okay, all right. So once we have this PRPP, so that ribose 5-phosphate is activated by ATP to form this PRPP. Okay. Okay. So once we have this PRPP, it reacts with glutamine, okay, and it forms this 5-phosphobeta-diribosyl amine. Okay, look what happened at this reaction. So basically, this amino group of glutamine is donated to this carbon 1 of PRPP. So it replaces, okay, this hydrogen with the amino group, okay, to form this 5-phosphobeta-ribosyl amine. Okay, 5-phosphoribosyl amine. So basically, it started to form the purine atom N9. Okay, if I'm not mistaken, N9. Okay, all right. Oh, okay. Another explanation that we can have at this step is the displacement of pyrophosphate group with amino group. Okay, saya mistake tadi. If you look at the structure of PRPP and 5-phosphobeta-ribosyl amine, we can explain that the addition of glutamine here, it replaces, okay, the amino group of glutamine is replaced the pyrophosphate group, okay, to form 5-phosphoribosyl amine. Okay, All right. Okay, the second reaction, okay, next reaction, okay, then this 5-phosphor beta deribosylamine is react with glycine. Okay, so there is an addition of atom from glycine, and this reaction is require of ATP, so which ATP is consumed here and it form glycine amide ribonucleotide. So kat sini dia dah build up, okay, the structure, okay, dah build up the structure, right? So this is the acquisition of purine atoms carbon 4, 5 and 7. Dekat sini akan adanya sebab there are three addition of three atoms addition from glycine. So it involves the formation of carbon 4, 5 and 7 of purine. Okay. So the glycine carbonyl group, okay, carbonyl group forms an amide. Dia akan forms amide with the amino group of phosphoribosylamine. Kat sini what happened here is the formation amide group, okay. Carboxyl group react dengan amino group, so it form this amide group, amide link, okay. Okay, with the form glycine amide. Sebab tu ada kat sini glycine amide ribo. So what kind of bonding involved between this glycine attachment carbon, okay, with the amino group of phosphorylamine. So you form glycine amide ribonucleotide. Okay, carboxyl group of glycine react with the amino group of phosphoribosylamine, and it, the attachment is linked by amide link. Okay, okay, alright. Ada soalan? So this is ATP consuming step. It require ATP of three atoms, okay, from glycine. Okay, okay semua. Alright. Next is the reaction of glycine amide ribo. Okay, formyl tetrahydrofolate. Okay, to form this. So dekat sini dia adalah the acquisition of purine atom carbon 8. Okay, okay. So kat sini carbon 5, 4, 5, 7. Sini carbon 8. Okay, kat sini kat N9. So you kena tahu dia dekat mana dulu. N9, it starts with N9 followed by carbon 4, 5, 7. Okay, dekat sini. Okay, then kat sini it form carbon 8. Okay. So what happened here is so if you look, okay, this free amino group kat sini kan, free amino group, okay, free amino group is formulated kan, dia by formyl tetrahydrofolate. So this amino group is formulated, okay, to yield this formyl glycine amide ribonucleotide. Okay, the amino group formulated by tetraformyl hydrofolate to form this FGAR. Okay. Alright, so formyl tetrahydrofolate so basically act as a formyl donor. So it react, okay, this formulate the amino group to form formyl glycine amide. Okay, so kat sini dah formation carbon 8. Alright, start with N9, then carbon 4, 5, 7, followed by carbon 8. Okay, next. Okay, formyl glycine amide ribonucleotide react with another glutamine. Tadi dah ada glutamine kan? Okay, another glutamine, and kat sini ada juga ATP. ATP is required, is needed. Okay, to form this formyl glycine amidine ribonucleotide. So what okay, kalau if we see glutamine here, we know that it contribute amino group and nitrogen. Okay, so what happened here? Dekat sini dia dah it start to build up N3. Okay, so the amide amino group, okay, the amide amino group of second glutamine is transferred, okay, to the growing, okay, to the growing purine ring to form formyl glycine amidine. So nitrogen from glutamine is transferred, okay, transferred kat sini. Okay, that's why it form dia akan bentuk formyl glycine amidine. So carbon, dia membentuk carbon 3 here. N3, sorry, N3. Okay. Alright. Then formyl glycine amide ribonucleotide, okay, undergoes, okay, and this reaction require energy. Okay, involve the usage of ATP, and it results in the ring closure that produce 5-membered imidazole ring of purine nucleotide. Okay, and it results in the formation of AIR, amino imidazole ribonucleotide. So this is the formation of imidazole ring. Okay, and this reaction require ATP. Okay. Okay. Ada masalah? So we have discussed the first 5 steps. Okay. Start from N9, carbon 4, 5, carbon 4, 5, 7. Carbon 4, 5, 7, then carbon 8 and 3. Okay. Ada soalan?
Tak ada marah tu.
Okay ke?
Okey je.
Okay, hopefully by Wednesday, if you can have physical class, and saya summarize lagi lah. Okay, should be tak susah pun. Okay, if you understand the structure of purine, okay, if you can remember in terms of the location of the structure, so that by having this pathway, you boleh tahu. Okay, it starts with the formation of the build up of N9, then followed by N9 from where? From glutamine. Then it build up carbon 4, 5, 7 from glycine. Okay, then followed by carbon 8 from tetrahydrofolate. N3 from N3 dari mana tadi? Entry from glutamine, for example. So kalau if you can at least imagine the structure of purine nucleotide, okay, so that you can relate, okay, what happened, okay, whatever happened in the pathway. All right. Okay, from AIR, okay, amino imidazole ribonucleotide. So we have obtained the imidazole ring. So now we are left with 6-membered nitrogen containing ring. Kan? Okay, kita ada 5 carbon imidazole, also 6-membered nitrogen containing ring. Okay. Okay, from AIR, okay, so we have this, it react with bicarbonate or carbon dioxide, and ATP require reaction. Okay, it use ATP. So kat sini we want to build up the carbon 6. Okay, so 3 of 6 atoms needed for the second ring in the purine structure are in place. Okay, carboxyl group is first added. Okay, so from bicarbonate, so it kita tengok kat sini this is structure of AIR. So it started to form the addition of carboxyl group. Okay, So this is from bicarbonate. Okay, this carboxyl group from bicarbonate. Okay, then this is carbon 6. We would like to form carbon 6. Okay, then it undergo rearrangement. Okay, so this CAIR, so carboxy amino imidazole ribonucleotide, undergo rearrangement and it transfer the carboxylate from the acyclic amino group to position 4 imidazole. So it undergo rearrangement, so it transfer this carboxylate group, this carboxylate to the amino group, ditransfer kat sini. Okay, ditransfer dekat sini. Ditransfer dekat sini. Okay, to the position 4 of the imidazole ring and form carboxy amino imidazole ribonucleotide. Okay. Okay, so basically formation ni adalah from AIR we want to build up CAIR. Okay, but it involves 2 steps. Okay, basically dia sama je. Okay, from bicarbonate ni, but it need to undergo rearrangement. Okay, rearrangement, dia transfer dekat sini. Okay, then the CAIR undergo reaction with aspartate. So aspartate donates its amino group in 2 steps. Okay, formation of amide by elimination of the carbon skeleton aspartate. This in a way. Okay. Okay, what happened here is okay, purine. Okay, let's look what happened to this. Okay, aspartate. Okay, this structure of aspartate berapa carbon if you remember? How many carbon? So it's a 4 carbon compound. Betul tak? Okay, from aspartate. Okay, so aspartate, what happened here is it donates its amino group in 2 steps. Okay, one in the formation of amide bond followed by elimination of the carbon skeleton aspartate. Okay, dia nak donate amino group je. Okay, so first of all, it involves this okay, so it donates its amino group. Okay, then to form the cycle. Okay, succinyl 5 amino imidazole 4 carboxamide ribonucleotide. So the succinyl we know it is a 4 carbon. Aspartate berapa carbon? Nak lagi tak?
Oh,
4 carbon. So basically, this formation, it involves hydrolysis of ATP. Okay, and is a condensation reaction, is amide forming condensation reaction, and it donates amino group, but it also okay, so it donates amino group dulu. Okay, it donates amino group dulu. Okay, and it's known as a succinyl. Okay, for carbon. Okay, so later dia akan ada elimination of this carbon skeleton of aspartate. Okay, then the cycle dia akan react with fumarate. Okay, fumarate. Okay, to form amino imidazole 4 carboxamide. Okay, ribonucleotide. So here what happened here is the elimination of fumarate. Okay, dia dah eliminate. Okay, the eliminate of fumarate. Okay, and from this ICAR. Okay, amino imidazole 4 carboxamide ribonucleotide. Okay, last one is the final carbon is contributed the formyl tetrahydrofolate. Okay, so it is it contribute the final carbon. Okay, contribute the final carbon. Okay, then last one it undergo hydrolysis to close the ring, the second ring. Okay, So to yield the second fused ring of the purine nucleotide. Basically, it involves 11 of log steps. Okay, yang first step tu activation of ribose phosphate to form PRPP. So what I would like you to do is go back, okay, try to understand the structure of purine. Sini kan, kita tengok balik kat sini kan. Go back and look at this diagram, identify, then based on this, okay, to go through this pathway and tengok dekat mana, okay, yang ring mana yang akan build up dulu. Okay, it started with N9, contoh N9 kan, lepas N9 dia akan start carbon 4, 5, 7. Alright, carbon 4, 5, 7. Then later carbon N3. Then you try to understand the pathway by looking at this diagram. So that you can see, okay, reaction 1 kat sini, reaction 2, okay, dengan glycine 4, 4, 5, 6, 4, 5, 7 ke something macam tu, 4, 5, 7, then N3, okay, dengan glycine kat mana lagi. Okay, so you have to understand every reactions in the formation of nucleotide of purine by looking at that diagram. Okay, tak susah pun. Alright, at least you understand how every atoms in the purine nucleotides how they are contributed by their sources. Okay, contoh glutamin bagi apa? Amino group. Okay, tetrahydrofolate. So what does tetrahydrofolate contribute to the structure? Okay, so bicarbonate contribute to which structure? Is it at carbon 6 or carbon 7? Or okay, you should at least try that. Okay, understand that. Okay, okay, ada soalan? Ada soalan tak?
Ada doktor.
Tak ada. Okay. Alright. So, okay. So, let's. Okay. So, this last slide. Okay, we last reaction. Okay, from 5-car ni, it anhydrolysis, so it forms inosinate or inosine monophosphate. The IMP is a very important intermediate in the formation of AMP and also ATP. Okay, so from inosinate, from IMP, let's look on how biosynthesis of AMP or GMP can occur from the inosinate. From IMP to AMP, it undergoes two steps of reaction. One is catalyzed by adenylosuccinate synthesis to form adenylosuccinate, which at this reaction, it requires aspartate and also GDP. Okay, so from adenylosuccinate, you tengok from inosinate structure, these two ring structure. So it aspartate contribute involved in this reaction. So it forms adenylosuccinate, and adenylosuccinate undergo the breakdown by the adenylosuccinate layers, where it eliminate fumarate. Okay, it removes this structure to form adenylate. So the phosphate comes from GTP. Nampak tak kat sini? Okay. Right. Okay, next from IMP, To form GMP, it also involves two steps reaction, but the intermediate is different. Okay, so to form GMP, it produces xanthylate XMP, where this reaction involves cofactor, which is NAD plus, and it involves a molecule of water, okay, that catalyzed by AMP dehydrogenase. Okay, AMP dehydrogenase to form xanthylate XMP. Okay, then XMP it form GMP, okay, which it involve the addition of glutamine to form XMP glutamine aminotransferase, and this reaction require ATP. Okay, so from IMP it can synthesize AMP and also GMP. Okay, ada soalan setakat ni? Okay, I'll stop here and I will continue next Wednesday. I hope we can have physical lecture next Wednesday. Kita akan, I'll try to recap, okay, on the basis of nucleotide next Wednesday and continue with the lecture. Okay, ada soalan ke? Soalan tak? No? Okay, IMP is what exactly? IMP, inosine monophosphate. What is inosine monophosphate? What is IMP? Anyone? Okay, IMP is intermediate. Okay, it is intermediate. So it in the purine biosynthesis, it produces IMP. Okay, IMP. It will not accumulate in cells, but it is rapidly converted to AMP and GMP and role in of nucleotide is it acts as an energy chemical carrier, kan, betul tak? So it is dia tak akan accumulate. Okay, this IMP dia as an intermediate in the formation of nucleotide, so it will not accumulate in the cells, but it is rapidly converted to AMP and GTP, AMP and GMP. Why is it important to have this AMP and GMP? It's part of your energy carrier. Okay, dia tak akan accumulate dalam cells. Okay, but it is converted to this because we know that AMP and GMP is very important as an energy carrier. Okay, alright. Ada soalan lagi? No? Silence semua kelas ni. Okay, if there is no questions, so I'll start yang tanya. Okay, what about practical? Okay, I will upload the apa ya? Practical punya amali, punya apa ya? Okay, so coming this week, okay, please wait, okay, get ready and read about and read on the protocol so that when you come to the lab, you are ready, you know what you need to do. Okay, I'm going to quickly discuss on the experiment, on the protocol by the next Wednesday. Okay. All right. So if there is no other question, thank you very much. Abu. Okay.
Thank you, Doctor.
Thank you, Doctor. Thank you, Doctor.
Doktor.
Thank you, Doctor.
Allah, baru keluar suara, ya Allah. Thank you everyone.